All right, so I think we left off somewhere around here with the idea that when you think about the microbes' perspective, they have quite the architecture to deal with when you look at the plant body, especially plant leaves. You can imagine a lot of microbes blow in across wing currents and manage to get onto leaves that way. Raindrops, snow, sleet, all can contain spores and microbes that land on the leaves, splash up from the soil, can cause the leaves to get colonized, as well as just when you have the seed germinating in the soil and the seedling is pushing through the soil to get to the surface. Usually, especially if you think about those seeds that are buried a little bit, some seeds, granted, are on the surface, but a lot of seeds are somewhat under the soil surface. And so as they're, excuse me, as they're trying to get to the sun, they're of course pushing through the soil and they can get colonized that way as well. Boy, sorry, I haven't talked much yet this morning. So here you're looking at the surface of a leaf with all the beautiful trichomes coming off the surface, which are just epidermal cells that have been extended out. And there's all kinds of different morphologies of trichomes. Then don't forget, we were discussing the idea that the stomata on the leaves, the pores, are often, hopefully, open during the day to take in CO2 and do photosynthesis. So then that perspective too, if you look at the scaling difference between the size of the pore and the size of a microbe, you can see that they can easily fit through the pores. So that's another way that we'll talk about next, the endophyte idea. That's one way that microbes that are on the surface can become endophytes and live within the plant body. Unfortunately, this one is Pseudomonas syringae, which is a pathogen. And so its intention is to get in and then start to attack. But there's a whole bunch of other microbes that are going in and benefiting the plant. And then here we're looking at a nicely stained, fluorescently stained leaf surface. The red is chlorophyll, but the green and yellow are microbes showing up. And you can see that it's patchy. It's a patchy distribution. It's not just this even homogeneous layer across the surface. They tend to congregate where there's more excretions going on. And so there'll be that patchiness. And you can see 1 times 10 to the 6th, 1 times 10 to the 7th densities per square centimeter. That's pretty decent densities. And again, here you can see them kind of congregating in cracks and crevices where you have some shelter, a little bit of protection from ultraviolet light. This is actually a cotyledon, which is the seed leaf, so to speak. Some seeds have big plump cotyledons, which are modified leaves that are just packed full of nutrition to help the plant, the seedling, grow and establish. So there's been some studies on what are they doing on the surface. This is a mock-up trying to show a couple of ideas here. So the leaf surface does have a waxy covering called the cuticle. All right, so that is one way that a leaf can kind of create a boundary so that it's not super easy for microbes to go deeper down into the leaf tissue. So that cuticle can help provide a barrier. It's very waxy, and it also helps them from not losing too much water out of their body so that they can control water loss through their stomata and not just have it evaporating out through the whole leaf surface. Then they're trying to show that there's dispersal going on. So as the microbes land on the leaf, there will be some migration, and that can be passive, dealing with water drops perhaps and the flow of water across the leaf, or they could be mobile and forming, if there's a thin film of liquid, they will be able to move via flagella, or they have gliding capabilities, swimming, swarming. So all of that can come into play. They're trying to show that there's surfactants that the microbe can potentially release, and that helps them move and establish. And then they're showing nutrients from the plant going toward the microbes. So they're trying to show that you have this dynamic where the leaves are exuding certain substances that the microbes can take advantage of. So when the microbes on the surface, the beneficial microbes, they're not attacking the cells and trying to get to the nutrients. They're able to exist with what is being kind of exuded by the plant. Here they're trying to show that the microbe can encourage a little bit of that exuding in the sense that that IAA that you see being released. So this was a study that was done on certain microbes that are releasing IAA, which is indoleacetic acid. It's a hormone. It's a plant hormone. So it's kind of weird, but plant-associated microbes, many of them, can make plant hormones. So they've co-evolved with the plant long enough where the microbe can make plant hormones, excrete that out to the plant picks that up. And in this particular case, it's allowing for the plant to release more nutrients so that the microbe gets fed in a friendly way. The microbe's not attacking the plant. It's just encouraging the release of some nutrients, some growth by releasing that hormone. And so there's many bacteria and fungi that are known to do this. They can release plant hormones. So those are the ones that have co-evolved with the plants long enough where that adaptation has established. They're also trying to show migration into nooks and crannies to try to help with dealing with, if you're a microbe on the surface of a leaf, you might have to deal with ultraviolet light, desiccation, free radical formation. So here they're trying to show congregation of cells, microbe cells, and they're trying to show this idea of a biofilm. So the EPS on here and quorum sensing is looking at the idea that now the microbes have established there's a population there. They're doing quorum sensing with each other. They're establishing a biofilm. And again, if you are in a wide open habitat like that, where ultraviolet could be beating down on you, or lack of moisture, it makes sense to go ahead and form a biofilm and try to protect yourself from all of that potential damage. So the beneficial microbes are interacting with the plant, but it's a more gentle, friendly interaction. They're able to get fed. They're able to release chemicals back and forth. So there is some communication going on between the plant and the microbes. So just like we're covered by microbes, plants are as well. And many of those microbes are beneficial to us and to plants. So you really don't want to have the surface of your body devoid of microbes. That's not a good idea, because then it allows pathogens an easier time of establishing on you and potentially getting in you. And so you want a community of microbes on you. And when you shower or bathe, you do knock some of those off with bathing, with soap. You certainly knock down populations, but not completely. The only way to completely rid yourself of microbes is if you burn your skin. And of course, that's not a good idea. But that's really the only way. You can't scrub your way free of microbes, and you shouldn't. You need that community on you. So plants are the same. The beneficials there can provide protection. They're not really showing that here, but there can be that protection of if an incoming pathogen lands on the leaf, if there's an already established beneficial community, the pathogen has a harder time. So that's another thing that can be good for the plant to have these epiphytes. There's some special circumstances that they've discovered in terms of some of the not so nice epiphytes. So this, again, Pseudomonas syringae, we already looked at it in that previous image where it was able to get into the stomata. So this one has a famous past in terms of the research on it, because it has the ability to create ice crystals on the leaf by excreting a protein. And it will allow for ice nucleation and an ice crystal to form even above freezing, if the air temperature is above freezing. And so this is kind of weird. And it's like, well, why would Pseudomonas go through the bother of doing this? Well, if you have ice crystals forming on leaf tissue, it tends to cause damage. The liquid to solid transition is expansion. It can cause tissue damage. The cold can cause tissue damage. You end up with cells starting to lyse open. And so for Pseudomonas, that's a pathogen and is way more aggressive than a beneficial microbe on the leaf. The lysis is what it's going for. It wants to break open the leaf cells so that it can get at all the nutrients inside the leaf and get that for its own needs. And this has been a huge problem in agricultural settings. We see that huge fields of plants can get wiped out with this, with Pseudomonas syringae sweeping through the fields. And so this caught the attention of plant pathologists, and they noticed, figured out what was going on. And then they said, well, okay, if we have all this ice nucleation going on by the Pseudomonas, what if we made a mutant of Pseudomonas that couldn't form that protein and couldn't make ice nucleation? And at a key stage of the plant's growth, we do a foliar spray with a solution of that mutant Pseudomonas. And so they did this, and this was quite a while ago, when they were doing this, and it caught the attention because this was probably the first example of a genetically modified organism getting sprayed out into, a natural setting. And so that created quite a bit of hubbub and a lot of debate about whether we should be doing that. The study was successful. They were able to show that the mutant Pseudomonas, if they sprayed it before the pathogen had gained a foothold, that mutant created a barrier, so to speak. It out-colonized the Pseudomonas, the pathogen Pseudomonas, and was able to keep the pathogen at bay and create far less damage. And so it was a success, but it created a lot of debate. And so that was one of the kickoffs of people becoming aware of the genetic modification idea and the whole debate on whether that should be done or not, which is a whole nother topic that we can't get into right now. But so yeah, it's an interesting history. And then this doesn't get talked about very much. In fact, when I was doing some literature searching on this, there's really not a whole lot published on this at all. And like nothing I could find within the last 4 years. But basically, in a nutshell, what they find is some conifer trees on the leaf. So phylosphere is referring on the leaf. And then some other plants like mulberry or partridge berry, and then some tropical species, of course, that we don't have here. But basically, what you can see is that there will be bacteria, nitrogen-fixing bacteria, hanging out on the leaves, and they'll form nodules on the leaves instead of the ones that form nodules on the roots. And when they were looking at that, at first they, were wondering, is that a pathogen hanging out on the leaves causing problems? And when they went to study it more, they realized, no, those are actual nodules. And when they peered inside and stained it, they could see that it was a bunch of bacteria packed in there. And when they, studied it more carefully, they've realized those bacteria were doing nitrogen fixation. And so this is kind of a rare case. By and large, the vast majority of information on nitrogen fixation is all about roots, and that the soil bacteria hang out with the roots, get into the root tissue, and into the nodule area and start fixing nitrogen. Well, in this case, it's leaf tissue that gets colonized, a leaf nodule forms, and inside of that nodule, you'll have the nitrogen fixers. So that's a pretty unusual, not widespread concept that, again, it's hard to find anything published on that lately. So here's a kind of a summary table of looking at microbes on the plant body on the surface. And this is kind of a summary of the big picture ideas. They mentioned both the good and the bad microbes here too. So this is a good summary table to think over and study. They mentioned the surfactants and auxins, that's the IAA idea that is allowing for nutrient release and letting the microbe persist. But yeah, patchiness, lots of environmental variables that they have to deal with. Okay, so with epiphyte research, I would wager that the epiphyte research started before endophyte research, and they knew more about epiphytes than endophytes for sure early on. But now that they know about the endophytes, and that's actually largely because of the human microbiome research, all of that kicking off really captured the attention of the plant biologists who said, you know, I wonder if there's a bunch of microbes living in the plant tissue, just like there are in humans. And so the field of endophyte research really took off. And now I would wager to say it's even more heavily studied than epiphyte research. And this is the idea now. Typically, when you say endophytes, you're more referring to the beneficial microbes, not the pathogens. And the idea that they are living in the tissue of the plant. So you have to imagine a plant seed, you know, germinating and growing. We already talked about the idea that you could get microbes going into the plant via the stomata. So here again is another excellent image showing that those rod-shaped bacteria sneaking into the stomata. And so certainly endophytes could establish via that mechanism. But there are other ideas too of how microbes get into the tissue in a friendly way without causing damage. So here's another image of a plant leaf on a tissue culture agar medium. And you can see all this fungal hyphae starting to grow out of the leaf tissue. So in tissue culture, this is one thing that you hope doesn't happen to any large extent, because when you're cloning plants, you don't want the plant to get overwhelmed by overgrowth of the fungus or bacteria or whatever endophyte. And so that's always tricky to establish a pure culture of the plant without its associated endophytes coming out and colonizing and sometimes overwhelming. So if you see tissue culture, one way you can tell that it's an endophyte, of course, is that there's no contamination in the area away from the tissue on the agar medium. It's all around the tissue and coming out of the tissue. Usually though, that shows up about 48 hours after you do a tissue culture experiment. And so if you see no contamination for the first couple of days, and then all of a sudden you start to see contamination, chances are it's an endophyte that's sneaking out of the tissue. And so endophyte, by definition, they live in the plant body, in the tissue. And it can be fungi, it can be bacteria, viruses too, but they know far less about the virus, the virome, than they do bacteria and fungi. Typically, it's a win-win where the endophyte is providing a service to the plant and the plant is providing a service to the microbe. Lots of interest in looking at, are they helping the plant defend themselves? So are they good biocontrol agents from that perspective? And also helping them deal with herbivores that come along and start munching on the plant. So when they were first noticing certain things just out in the field, one of the earlier observations was they would watch cattle feeding in their grazing land, and they noticed that cattle would avoid certain native grasses. They wouldn't chew on them. And they started to wonder, why were those plants being left alone and everything else is getting chewed on? And so they went to that plant and started looking more carefully, and they found certain fungi in these grasses. So those were really the first endophytes that were discovered to any serious extent, and actually studied more carefully. And so the grass family is Poaceae. And here you're seeing a leaf surface or a leaf cells, and you can see the fungal hyphae staining the dark blue. This is a methylene blue staining where the fungal hyphae pick up on that stain, and you can see it colonizing in the tissue of the leaf. So tall fescue were some of the plants, and ryegrass were being studied for this, and they discovered this fungus, Acremonium species. There's a couple different species. And when they started looking at that fungus more carefully, it was producing some pretty nasty chemicals, ergopeptides, and others that are considered toxins to mammals, including humans. And so that was why the cattle had learned not to chew on those plants. It was dangerous to do so. When they started looking at what else can it attack besides mammals, they realized those chemicals are also effective against the nematodes and aphids, variety of different insects too. So nematodes are just little tiny worms that hang out primarily in the soil. And by the way, there's both good nematodes and bad nematodes from the plant's perspective. There are some nematodes that are good biocontrol agents, actually, but there are some nasty nematodes as well. Aphids are always nasty. Really don't like those things. They cause problems in our greenhouse. When they started working with this fungus, they realized they were having a heck of a time growing it. They could do all kinds of extractions on it and figure out what chemicals it was producing, but getting it to grow to any abundance, they were really having a hard time. And so that really got them confused. It's like, okay, how in the world is this fungus surviving then? Like they thought it was a soil fungus and somehow getting into the plant through a mechanism through the roots or something. And they didn't see that ability in controlled studies. So basically, when they started looking more carefully at the seeds of these grasses, they could find that it was in the seed. It was packaged in the seed. And so as the mother plant, so to speak, is making those seeds, the fungus can actually mobilize to this developing seed and get encased into the seed tissue before the seed is mature. And so it's a package deal. When that seed gets released, the fungus is with it. And so that's the way that it goes from plant to plant. It's not free living, that at least that the scientists could find. They couldn't see that it was a free living. So that is saying this is a pretty tight relationship if you have that fungus pretty incapable of surviving on its own and is transmitted via the seed route. Then another group of endophytes that were early on studied were some bacteria. They're called PPFMs, pink pigmented facultative methylotrophs. So these bacteria were actually discovered quite a while ago and by plant physiologists. And it's a methylobacterium species. And again, pretty tight relationship. They only really see it seed transmitted. They don't really notice it in the soil system and or on the leaf surface. They can see it in a bunch of different plants. So it's pretty widespread, wide host range. Gymnosperms, angiosperms, non-vascular plants, vascular plants. So it's a pretty common group. So they grow pretty slowly though. And in order to detect them, you need specialized media because they're kind of picky if you try to grow them in the lab setting. And so for the longest time, they were difficult to detect. And I remember reading one paper by a plant physiologist who was talking about these microbes, realizing now, way after the fact of, boy, these bacteria have been here with the plants, with many, many plants. It's ubiquitous, so really common. never detected. And so all these decades of plant physiology research kind of went up in the air in the sense of endophytes are known to produce all kinds of chemicals. So now the question is, when they are talking about all these chemicals that the plant is making, how do they know it's the plant making it and not the endophyte? And so a lot of plant physiologists kind of groaned at this. It's like, oh no, now we have to think about all these endophytes and all the stuff they're doing. And maybe what we attributed to the plant, it really isn't the plant, it's the endophyte. And so it kind of put a wrench in the plant physiologist world of what they were saying about what the plant is capable of. And now there's this idea more firmly established that we need to pause here and think, is it the plant really doing it or is it the endophyte doing it? And so it's actually an exciting area of research, but I could see how the early plant physiologists are like, oh, are you kidding me? Now we have to rethink what the plant is capable of, because maybe it's the endophyte doing this. So anyways, methylbacterium, it actually can take any methanol that might be being produced as a waste product and use that and break it down. And so that's good for the plant. And then it also is excreting plant growth factors like hormones and other chemicals that the plant can use. And so they're looking at it too to see if maybe it's just it's so common throughout broad host range because it's doing a lot of different things for the plant, not just removing undesirable waste products that the plant is producing, but maybe it's protecting the plants too in some way, yeah, kind of like a biocontrol agent. And it's getting fed by the plant, so it's persisting. And so they suspect it's a win-win. And again, was kind of one of the early group of bacteria that were discovered to be endophytes and common endophytes. So getting into the plant. So you have to imagine that could be a little tricky because plants are pretty well defended, more so than we might be in some regard, in the sense that because they can't get up and walk away from danger or from organisms trying to attack them, they have pretty sophisticated mechanisms in play. One of the largest or most accepted hypotheses right now is that most endophytes are gaining entry through the roots, whether it be through root hairs, where those are just modified epidermal cells. So that might be an easy way for them to gain entry is through the root hair system. Or maybe some damage. If there's a little nick or abrasion where they could get in. And so that's a common thought, is that a lot of the endophytes that they see in the plant body originated in the soil and are getting in through that way. And so they're trying to show this. They certainly know that root surfaces are super well colonized by microbes. And so it's not too much of a stretch to think of them being able to get in. Soil is pretty abrasive as the roots are moving through. That friction can cause some slight damage. And again, the scale issue doesn't take much for a small size or crack to open up for the microbe to be able to get in.
But the bacteria and fungi, they're like co-evolved, so like can't be like free living anymore. Did they like enter the same way and then eventually like their metabolism just
Yeah, they suspect that's probably what happened way back. And then like the ones that get transmitted via seeds, probably
like
way back, they got in and then they adapted so well to the plant. Now they can't exist outside the plant type idea. Yeah, that's what they think. And then there's, excuse me, there's endophytes that will get into the plant body and they're not quite that established yet to the point of being able to only exist with the plant tissue. Some are like that, but many are not. Many can live on their own, and then they can go endophytic as well. And so that's got a lot of people thinking about, using endophytes, using bacteria to see if they can go endophytic in order to help the plant. So that's one way that they introduce bacteria and then they follow and track to see if it can go inside the plant tissue. They'll put them into the soil and see what happens. The other idea is, again, doing foliar sprays. You can also introduce microbes that way too, deliberately, or, you can see it happening naturally as you do research experiments. So here you're looking at fungal hyphae, excuse me, and you can see that the hyphae are indeed finding the stomata here on the leaf surface and getting in that way. So both, beneficial and detrimental. The pathogens can do this too. It's not just beneficials. But most scientists seem to think the vast majority of endophytes are coming from the soil. That if you just think about the sheer numbers and diversity of microbes in the soil, that it's easier to see more possibilities of endophytes through the soil route. And then we already talked about how some are totally just seed transmitted. They can't live, they don't, at least from studies being done, they don't seem to be able to live on their own anymore. So they are more tightly, they have a long coevolutionary history with their host plant to the point where this dynamic has been set up so that the endophyte is in the seed, and as the seed is germinating, then it can mobilize with the developing shoot and go into the shoot and leaf tissue. So here, this statement here, the endophyte grows up the stem and into the seed head. Okay, so that's getting at the idea that it can move through the developing plant body as the plant is growing. That fungus we talked about is capable of migrating. And so they're looking at this more carefully when they discovered these grasses having seed endophytes, and they started looking at a bunch of different seeds. Here's one on cannabis, looking at seed-borne endophytes inside. These are bacilli, so this is bacteria that are known to inhibit certain fungal pathogens. So here, Alternaria, Penicillium, Cladosporium, these are all Aspergillus, Rhizopus, Fusarium. Again, those are all pathogens, and they are finding that the seed comes packaged with biocontrol agents, endophyte biocontrol agents in the seed. And so there's a pretty big push right now to look at seed endophytes. And then there's another group of scientists that are starting to question the routine idea when you get seeds. Often what you do is you surface sterilize the seeds. And they're starting to question that. Like, is that really what we should be doing? Because maybe those surface microbes are beneficial, and here we are wiping them out. And are we doing more harm than good? Initially it was done out of fear that the seed might be coated with pathogenic microbes. And so they wanted to, get rid of them, which is understandable. But now with more awareness that the plant is super well colonized by beneficials as well, and that the seed certainly has beneficial endophytes inside, now they're starting to question, maybe we shouldn't be killing off all the microbes that are on the surface of the seed, and that we could be really harming the plant's ability to germinate well and be thriving right from the get-go, because it's lost some of its partners through us killing the surface microbes. So there's a lot of talk about that right now too. And I just wanted to make sure that you understood that, yeah, definitely endophytes can move through the plant body. They're not stuck necessarily right where they gain entry. And so it depends on the microbe. Some may stay put, but some travel through the plant body. This is some nice green fluorescent protein staining of bacteria that are starting. So A is the lateral root, And you can see they're gaining entry through a lateral root and then also through the root hairs. And then getting into the root tissue proper. And then getting into the base of the stem of the rice plant. And then E and F, those images down low, E, F, and G are looking at leaf tissue. And so definitely they can track that, yes, some microbes definitely do migrate. And then, of course, rice forms the grains, on the surface up in the tips of the leaves. So perhaps some of them are getting into the developing seeds as well. So they don't have to stay put necessarily. Here's another different plant. This is wheat embryo. So this is in the seed. These are Streptomyces bacteria that have gotten into the seed tissue. And it looks like figure A is just within the first 24 hours and figure B is after 3 days. And you can see that their populations are increasing. So they're getting established and growing. And here as well. So inside the seed. And Streptomyces are famous for being in the soil. So they've managed to go from the soil into the plant and then make it to the developing seed. Okay. I suppose I should stop here. I wanted to go over the essay portion. If you have EduCat open,